This experiment we uses suspension cells as an example. Step 1. Prepare the sample. Step 2. Centrifuge at 12,000 rpm for 1 minute. If adherent cells are extracted, they should be digested with trypsin and pipetted. After centrifugation and supernatant centrifugation, we got cell pellets. Step 3. Discard the supernatant. Keep cell pellets. To add 200 microliters buffer to cell pellets, and shake it until completely resustained. For cells and tissues samples, we recommended you to add 4 microliters RNA enzyme solution. RNA enzyme solution is purchased separately. This experimental sample is a cell sample. Therefore, this step requires the addition of 4 microliters of RNA enzyme. Shake it for 15 seconds. After shaking, let stay at room temperature for 5 minutes. Add 20 microliters proteinase K. Mix well. If the sample is animal tissue, incubate it at 56 degrees Celsius until the tissue is completely dissolved. Centrifuge briefly to collect the solution on the inner wall of the cap, and then go to the next step. Add 200 buffer delp. Mix slowly by inverting the tube. Incubate at 70 degrees Celsius for 10 minutes. A white precipitate may be produced when buffer DLB is added, which will generally disappear when incubated at 70 degrees Celsius and will not affect subsequent experiments. And centrifuge briefly to collect the solution on the inner wall of the cap. Add 200 microliter anhydrous ethanol. Mix well. Precipitation may occur at this point. Prepare the corresponding number of spin columns GHC and 2 ml collection tubes. The spin columns GHC is placed in the collection tube. Transfer all the solution with precipitate to the spin columns GHC. Centrifuge at 12,000 rpm for 30 seconds. Discard the flow-through, and put the adsorption column back into the collection tube. Add 500 microliter buffer DWE to the adsorption column. Notice, please make sure to add the appropriate volume of anhydrous ethanol as assigned before using buffer DW1. Centrifuge at 12,000 rpm for 30 seconds. Discard the flow-through, and put the adsorption column back into the collection tube. Add 500 microliters buffer DWR+ to the adsorption column. Please make sure to add the appropriate volume of absolute ethanol as assigned before using buffer DW2+. Centrifuge at 12,000 rpm for 30 seconds. Discard the flow-through and put the adsorption column back into the collection tube. Repeat the previous step. Centrifuge at 12,000 rpm for 3 minutes. Discard the flow-through. Prepare to clean 1.5 ml centrifuge tubes. Transfer the adsorption column to a new 1.5 ml microcentrifuge tube and add 60 microliters buffer EB to the central area of the adsorption membrane. Stay at room temperature for 1 minute. Centrifuge at 12,000 rpm for 60 seconds. Discard the adsorption column. And collect the DNA solution in the centrifuge tube. Extraction ends.